Today, I would like to welcome Dr. Mithila Jugulam. She is a professor of agronomy at Kansas State University and a recipient of the 2021 Global Food Systems Seed Grant Award. The title of her presentation is Development of Herbicide-Tolerant Wheat, Valuable Resource for Improved Wheat Management in Kansas and Beyond. Dr. Jugulam?
Oh, thank you. Thank you very much for the introduction. And also, I would like to acknowledge the opportunity that I got from Global Food System Grant to do this important research. And as I explained, you see, this funding was really valuable for me to expand the research beyond what was done using this grant funds. And also, I'd like to acknowledge my co-PI, Dr. Asif Mohammed from Heartland Plant Innovations. Their contribution to this research is also very valuable for my group. So very briefly about why this research is important. So wheat, as we all know, is widely consumed grain across the globe and also one of the most important crops grown in Kansas, especially winter wheat. And as in other crops, weed control can be a problem in wheat as well, especially as you can see in these images, these wheat species shown in the wheat crop at the time of harvest, they're all herbicide resistant weeds. So that has been a challenge in the recent past, not only in wheat, in other crops. Evolution of resistance in weed species has been a challenge for management of weeds in crops such as wheat. So we need to have better management strategies that we need to explore for weed control. Also, it's obvious that we need to have timely and efficient weed control for increased yields of any crop, also particularly for wheat as well. And especially, as I mentioned earlier, in the event of development of herbicide resistance in weeds, it has become even more challenging. So some efforts have been made in developing herbicide-tolerant wheat, but these are not in terms of when you compare to other major crops such as corn or soybean, where we have many more herbicide-tolerant traits that have been developed compared to wheat, where we believe it is not gained a lot of attention compared to those other major crops. Having said that, recently we do have some acreage of wheat in terms of growing herbicide-tolerant varieties, such as labeled as, or they are sold as Clearfield wheat or coaxium wheat. So these wheat varieties are very specific to a particular chemistry that they are tolerant to. In other words, if you use these varieties, you can control the weeds using certain chemistry of herbicides. The downside of this technology that has been released and widely commercialized is that many weeds in our wheat cropping system have already evolved resistant to these two chemistries. Therefore, we need more options to explore development of herbicide-tolerant wheat variety. In this regard, we did some preliminary work and using herbicides such as protoporphyrinogen oxidase, in short, PPO inhibitors. These are herbicides. I gave some examples here, lactofen and fomesafen, or the chemical names of these herbicides. For lactofen, Cobra is the commercial name, and for fomesafen, it's Reflex. That's the trade name of these herbicides. They are very selective herbicides, and they can control many broadleaf and grass weeds. Luckily, we don't have reports of weeds that are resistant to these herbicides in wheat crops. So we thought this is a good option for us to explore and see if wheat varieties resistant to this herbicide can be developed. The other thing is right now these herbicides are not registered for use in wheat, whereas they are used in soybean very extensively. And also we have done some preliminary work and found that there is some natural tolerance to lactofen or formicefin among wheat germplasm that we tested. And also having PPO inhibitor tolerant wheat varieties can help weed control throughout the wheat cropping season. So this is the advantage of herbicide tolerant technology, not only in wheat, in any other crop. Growers find this a easy way of controlling weed, like almost like a silver bullet. They can go and apply the herbicide anytime during the cropping season so that the crop is not affected and they can have better weed control. So I want to explain very briefly how these herbicide like PPO inhibitors actually kill the plant or how they work inside the plant. So basically these herbicides have a target site in protoporphyrinogen 9 biosynthetic pathway. I don't want to go over every step here, but this pathway is extremely valuable for plants for the production of heme or chlorophyll both, or heme eventually produces phytochrome, and chlorophyll obviously is the pigment that's extremely important for plant growth and development. During photosynthesis, the chlorophyll plays a major role. So what happens is that during the biosynthesis of these two pigments, chlorophyll or phytochrome, the photoporphyrin 9 needs to be converted to protoporphyrin 9 via action of the enzyme PPO, which is the target of these herbicides. Basically, the protoporphyrinogen oxidase is the enzyme. The herbicides, when we apply this herbicide, this enzyme is inhibited. The precursor or the previous step in this pathway accumulated, that is protoporphyrinogen 9 gets accumulated because downstream reactions are inhibited, and this accumulated for photoporphyrinogen from chloroplast, it gets moved into the cytoplasm. So in the cytoplasm, the protoporphyrinogen obviously goes through the steps of getting it converted to protoporphyrin 9. Only in chloroplast, if it is produced, you will see the downstream pathways occurring. Whereas since it is getting accumulated in cytoplasm, what happens is the protoporphyrinogen 9 reacts under light and also in the presence of oxygen. They produce singlet oxygen. Therefore, we see the lipid peroxidation and tissue necrosis. So basically, this cartoon shows what happens inside the chloroplast and cytoplasm once we inhibit this enzyme, and how the plant dies. Basically, plant dies because of oxidative degradation and lipid peroxidation, which is the membrane damage. That's how these herbicides kill the weeds. So with that in mind, we hypothesize and probably the natural variability present among the wheat varieties or in the germplasm can help us identify source of tolerance to these herbicides. And although the overall goal is to develop herbicide tolerant wheat so that we can have wheat control throughout the season. So our specific objectives were screen wide germplasm of wheat, including some winter wheat, spring wheat, and also EMS, which is ethyl methane sulfonate derived mutant lines, and see if we identify any tolerance to the PPO inhibitor herbicide. And then once we identify, we also wanted to characterize the level of tolerance, which is really important because in terms of developing herbicide tolerant crops, we need to have really high level, which means the wheat crop in this scenario or any herbicide tolerant crop should be able to withstand application of higher doses of herbicide than the normally recommended dose. So in this experiment or in this research, we used 24 winter wheat lines and also mutant lines. These are the lines provided by Heartland Plant Innovation Center. They routinely use this wheat germplasm in their research. We are fortunate that they were able to give us quite a bit of germplasm collection of wheat. So we used winter wheat, mutant lines, as well as spring wheat varieties. And these were grown in the greenhouse. And when they were at 4 to 5 leaf stage, wheat seedlings, we used them for herbicide treatment. This stage of wheat development was selected based on what is given in the wheat guide. Essentially, growers apply herbicides post application of herbicide only at this stage of wheat development. So we used various doses of lactofen or fomesafen. These are the two PPO inhibitors we selected to screen, because these are the widely used herbicides in this chemistry across cropping systems. So we used various doses, which is 0, obviously non-treated plants. And then what it means is 0.25, 0.51, 24, and 8 means these are the doses of herbicide below and above the field recommended dose, which is 1x. We usually, if you look at the herbicide label, the recommended dose is given as 1x, and which is 220 grams and 280 grams per hectare for lactofen and fomesafen respectively. And then after treating the seedlings with these herbicides, we record the injury, basically looking at the visual damage of the plant, and we rate them 0. 0 is no injury, and 100% injury is completely dead plants. So we do take these readings at various weeks after treatment, 1, 2, 3, 4 weeks after treatment. And after 4 weeks, we also collect the biomass to see the growth reduction after application of herbicide. So once we got the wheat, this is the data for the winter wheat. Once we treated this winter wheat 24 varieties that I showed with lactofen or fomesafen, we recorded the visual injury data as well as biomass. Here on the top graph, I'm showing the lactofen injury after 1 week of treatment, and below I'm showing the lactofen injury after 4 weeks of treatment. This is just, although like I said, we took the data for each week, like 1, 2, 3, and 4 weeks after treatment, just to show you here. Initially, after 1 week of treatment, you see some injury, like up to, for example, in the most tolerant one, we see up to 40-45% injury, and in the most sensitive one, it may be up to 70%, extremely sensitive. But the reason I'm showing here is initially you see the damage, and on this other side of the graph, you see we have the injury data at 1 week, 2 weeks, and 4 weeks after treatment. So although first week you see injury, but as the plant grows, the tolerant one come down in terms of injury percentage, which means they are recovering from the herbicide damage and growing normally. That is what is shown here. However, the sensitive one continue to show higher damage, and they eventually may not compete with the most tolerant ones. They won't produce enough biomass. So based on this visual injury data, we identified, we numbered them as W1 to W24. So W13 and W4 were found most tolerant and sensitive respectively to lactofen, whereas W1 and W24 were most tolerant and sensitive respectively to fomesafen herbicide, which is very important because we have 2 different varieties showing tolerance to 2 different herbicides, which helps a lot in terms of characterizing the resistant later on, and also transferring the resistant to desirable wheat cultivars. Another thing I want you to pay attention here is, as you see in the top graph, I showed W13 and W24, both are about the same level of approximately same level of injury, and also at 4 weeks after treatment, also the injury level is not so different. So if you look at this W24, although it's showing more tolerance to lactofen, and this one is extremely sensitive to fomesafen. This we call in wheat science as a negative cross resistance, which means the variety that is most tolerant to one herbicide is extremely sensitive to other herbicide. In terms of like a big picture thing, this gets very valuable for us. For example, if we develop lactofen resistant wheat, and if we end up having wheat species evolving resistant to that herbicide, at least we can use the option of switching it to other chemistry that is tolerant to wheat, so that those weeds can be controlled. So in a long run, I see really value for this result, like one negative cross resistant result that I just explained. For now, I thought this is really exciting result so that we can take advantage of it. So here are some images of wheat plants showing the response to lactofen or fomesafen. Like I said, the W4 is very sensitive to lactofen. You see, beyond 8x dose, the plants are killed. Whereas W13, even 16x, we see a little bit of growth, but eventually they come out of this also. But more importantly, if you look at field recommended dose of lactofen, or even up to 4x or 8x, they're doing very well compared to the sensitive one. Similarly, for fomesafen also, we have seen the sensitive one getting killed at 4x or even 2x, you see more damage, whereas the tolerant ones are doing well at 4x. But as you can see in these images, wheat seem to be more tolerant to lactofen than fomesafen in general. So again, the data shown here is from our dose response assay. Basically, in the dose response, we have used several doses of either lactofen or fomesafen and collected the biomass. This data comes from the biomass values. So basically, based on this dose response analysis, calculating the GR50, which is the growth reduction of 50% of the plant, the herbicide needed for this reduction of 50% of growth is called GR50. Based on that, if you see, the most tolerant one needs a very high amount of the herbicide. As you recall, the field recommended dose is 220, but here we need a lot more herbicide to reduce the growth by 50% compared to the sensitive one. Based on this analysis, we found the W13 is about 15 times, or had 15-fold tolerance to lactofen, whereas W1 has the 1.7-fold tolerance only. So like I said, lactofen, we seem to be more tolerant to lactofen than fomesafen. And we have also screened additionally 30 EMS mutant lines and 12 varieties of spring wheat as well. The similar testing was done, treating them with the herbicide, lactofen or fomesafen, and the response varied. But more importantly, we found that these mutant lines of spring wheat were more sensitive to PPO inhibitors than winter wheat. So the focus, as I explained in the previous slides, the winter wheat showed higher tolerance to this herbicide, and we want to focus more on that, on those genotypes. So basically, in terms of impact of this research, it helped us to identify resistance or tolerance to lactofen or fomesafen, which is really a step forward, so that we can use these lines to introgress the trait into commercially suitable wheat lines, and thereby we have opportunity to develop PPO inhibitor resistant wheat. what is currently we are doing is because of the opportunity that I got from Global Food System Grant, we kind of reached out to Kansas Wheat Commission. They are funding now part of this research. With this support, we are continuing the research, looking at mechanism of resistance or tolerance to PPO herbicide. Why are these genotype shows more tolerance compared to the sensitive one? And also we want to look at the genetic basis of this resistance so that we understand how we can introgress the trait. And one more important piece of this research, taking it to next level, is that we want to evaluate under field conditions how these lines will respond to lactofen or formosefen treatment. Like I mentioned, we received funding from Kansas Wheat, which is a big step forward in this research. And with that, I'd like to acknowledge again your support for this initial research, and Kansas Wheat, which is currently supporting. And my group, especially Aarti and Susi Sudhakar, they both worked on this project. That was great. And hopefully, with these results, I'm also looking at putting together a manuscript for publication. Aarti is working on that. Hopefully we get some publication out of this research. Any questions? Again, thank you for your attention.
Thank you so much, Dr. Jugulam. If there are questions, or if you would like to discuss this research further, please feel free to reach out to Dr. Jugulam. Again, she is in the Department of Agronomy at Kansas State University. Thank you.